Hello, everyone, and welcome back to the first session of OmicsLogic Introduction to Bioinformatics program. This program is an introduction to the field of bioinformatics or the intersection of bioinformatics and biology. In this program, we will discuss the changing landscape of data-driven research in life sciences and the opportunities that come with the widely accessible high-throughput technologies and computational methods for analysis and annotation of such detailed data in research and industry. We will discuss various emerging technologies and their impact on healthcare, agriculture, environmental sciences, public health, and more. My name is Beepsha Biswas, and I am the OmicsLogic Community Manager, responsible for daily interaction with thousands of users that are part of our bioinformatics community, managed by Pine Biotech. Let me start by introducing Pine Biotech, the company behind OmicsLogic, share with you details about the upcoming OmicsLogic programs, and explain how live training resources will be used in the Introduction to Bioinformatics program. Over the course of the past several years, our team has been collaborating with a number of academic institutions around the world that have participated in content evaluation, curriculum review, and project design. As a result, the OmicsLogic programs have been thoroughly evaluated and are known to lead to excellent outcomes. Importantly, the programs are designed to support students with minimal bioinformatics background, including biology and biomedical studies majors, as well as faculty interested in bioinformatics, data science, and biomedical research. The goal of the OmicsLogic programs is to provide training in bioinformatics, enabling independent research guided by mentors and peer example, developing a growing community with shared passion for data-driven research and appreciation of citizen science, enabling students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in life and data sciences. In 6 interactive sessions, you will get to see the broad applications of next-generation sequencing, biostatistics, and tool for big data analysis. In practical assignment, we will be learning about the logic of bioinformatics and see how data science methods can be used to study biology. In conclusion, you will also be able to plan how to take what you have learned and apply them to an independent research project using tools and methods we cover in the program. So this is the schedule for the 6 sessions that are going to be part of this 1-month program. And we will be discussing them in more details. And now let me introduce you to the main speakers of today's session and the mentors of this 1-month program. We have Ilya Brodsky, our Pine Biotech co-founder and CEO. In his current role, he has extensive experience working together with industry and academic partners that are applying multi-omics integration and bioinformatics to a variety of biotechnology and clinical applications. Dr. Harpreet Kaur is a certified OmicsLogic trainer and expert bioinformatician who will guide the hands-on sessions and assist the participants with projects and analysis during the program. Her area of specialization is cancer genomics and machine learning. Dr. Mohit Mazumdar is our OmicsLogic project mentor and an expert in big data computational analysis. Dr. Mazumdar has a PhD in computational biology and over 10 years of experience working with industry and academia. And now I would like to pass it on to Ilya to take over the session. Thank you, everybody.
Thank you, Bipsa, and welcome everyone to this first session that will be focused on the very basic introduction. So we'll talk today about processing and analysis of high throughput data sets and some of the logic behind analysis pipelines. We'll also discuss some of the resources that you have access to in this program and how you can combine the logic of bioinformatics with practical skills that are needed to succeed and excel in big data analysis. My name is Ilya Brodsky. I'm the co-founder and the CEO of Pine Biotech, and I'm excited to join you today for this short program. The program is just a first step, and it's designed for biologists, biology students, for those that are in clinical research, biomedical research, but also those that are interested in other aspects of next generation sequencing and big data in life sciences. So why so many of us are actually so interested in bioinformatics and data science? The truth is that biology is acquiring the character of a data science. So many experiments are producing large volumes of heterogeneous information that needs to be analyzed, processed, and integrated to lead to insights and discoveries. And so the goal of the OmicsLogic programs is to introduce biology as a data science, preparing biologists and clinicians for this influx of data that will only continue to grow, and also helping them master the associated methodologies of data-driven research by focusing on projects and understanding how bioinformatics can help us overcome the challenges of discovery and experimental design as we think about different aspects of life science research and industry. So in this session, I will introduce some of the topics that we're going to be covering. I'll talk a little bit about the basic bioinformatic approach to big data by showing you how to run and analyze the outputs from different pipelines. We'll talk about different types of omics data because there's a variety of different information that they can provide. And we'll also talk about some of the resources that you can leverage in an asynchronous way after this session and during this whole program. So the course that we're discussing here, this program and today's session, is really designed for those that come from a non-engineering background to introduce bioinformatics for students, biologists, and clinicians, and understand how you can personally get started with your bioinformatics research journey. So one of the important things that we have to recognize is that the influx of data is continuously increasing in speed, variety, and volume, all the way back from the 1950s when the genome was first, when the human genome was assembled, up until today. There have been new technologies like next generation sequencing that have produced continuously growing volumes of data of different types and have been made publicly accessible. And one of the primary examples of that is the National Center for Biotechnology Information. The National Center for Biotechnology Information contains different types of repositories, and one of them is the Gene Expression Omnibus, or GEO, which by itself contains over 80,000 publicly available datasets that include gene expression profiles of hundreds of tissue types in thousands of organisms across a variety of different types of conditions. So we really see how the available data opens up the door for those of us interested in handling these datasets and designing hypotheses driven by data that is already there, to be able to access high quality data sets that were produced by cutting edge labs and in amazing projects that we can all learn from and leverage for our own study and research. And that's what this program is going to help you do. We'll actually go on NCBI, GEO, SRA, GenBank, and other repositories, find project data, analyze that data together, and discuss the results, the different inputs and outputs of technical pipelines, but also conceptually we'll be able to look at the different types of analysis methods that are applied to different types of data. So what kind of data are we going to be talking about? We are going to be focusing on biomedical research as an example, but you will also be able to apply this to agriculture, biotechnology, and basic life science research. However, in the field of biomedical research, it's important to understand the critical role that biomarkers play in the diagnostic process, helping clinicians make day-to-day decisions about their patients, including characterizing the disease, identifying the stage of disease progression, and selecting therapeutics. This is based on the work of researchers that can produce high throughput data of different types to study what happens at the subcellular level. And importantly, to integrate that with all kinds of additional data sets and repositories that exist to describe the condition from a clinical perspective, as well as to identify the specific interactions between proteins, pathogens, and hosts, as well as the different types of processes that happen inside the cell. So this program is designed to highlight the role of large omics datasets, or big data, that is shaping modern biomedical innovation, where molecular data is used for precision diagnostics. Big data integration and machine learning are transforming personalization of treatment selection, and innovative algorithms allow for unprecedented capabilities with drug discovery, design of molecules, and drug repurposing. So hopefully we will start talking about all of these topics today. But before we do, let me ask you a few questions. So first of all, what are some examples of omics data that is used in research that you know of? Okay, so you can use the chat box to just write your answers, and later on we'll be using your responses to continue and speak about different aspects of the program. So what kinds of omics data are you familiar with? Vabihav, proteomics and genomics. Joshua Stephen is saying proteomics. Okay, Vibham Sharma is saying metabolomics, genomics, transcriptomics. Excellent. So a lot of you are familiar with the term omics. And for those of you that are putting in answers like SRA or other types of answers, really the question here is, SRA is a repository, it's a database. But here we're just talking about the different types of omics, interactomics, okay? So it's very interesting to consider how many different types of omics are going to be listed here. So that's really the point of this question. So thank you for answering this question. Let me ask you another question related to this. What kind of information can we learn from transcriptomic data? So Genova is saying gene expression, sequences, differential gene expression, mRNA, RNA detail interactions, mRNA gene genetic expression. Excellent. So let's think about transcriptomic data. So transcriptomic is one type of data that you all correctly identified. It speaks about the RNA, mRNA, different types of small RNAs like microRNAs, long non-coding RNAs. So transcriptomic is everything that is being transcribed, so everything that is being expressed. Now, studying gene expression data, we can look at levels of gene expression and compare them between different types of conditions. So that's called differential gene expression. We can look for profiles of gene expression across a variety of classes of conditions or samples. We can also talk about transcripts or isoforms, how genes can be alternatively spliced in the process of transcription, and identify differences in alternative splicing between different conditions. And obviously, depending on the data preparation protocol, we can also explore other types of RNA that is being expressed. So I think it's important to understand that we have multiple types of omics data, and each type of omics data can provide us with different types of information. Let me ask you another and final question for now. What are some of the challenges that you know of with analysis of omics data? Ishwarya is suggesting accuracy, mutation, data redundancy, not much exposure, precision, false positive, repeated unwanted expression, errors, size, missing out on some data type and accuracy overlapping. Okay, so there are some interesting challenges that are being mentioned right now in the chat box. And let's briefly summarize what is being said. So one aspect is technical. Technical, just the size of each data set. For example, when we talk about transcriptomic data, we talk about gigabytes or hundreds of megabytes per sample. The bigger the sample set, maybe tens or hundreds of samples, very easily we can see how our computers are going to run out of space and run out of computational power. Now, the other side of it is just the ability to annotate. So when we have a reference genome or the transcriptome, when we have the reference genes and their annotation, there's a lot that has to happen before we can interpret with confidence. And therefore, a large part of the analysis is being able to identify the correct annotation databases, the right references, and link all of that information together that the identified results could be tested biologically. Now, it's also important, what is described here is that we're dealing with real biological samples. So samples that are being studied in India or in China or in the US might be prepared in different ways, sequenced at different time points using different machines. So there is a large variability between the quality and the specific biological conditions that are being provided in a way that we are used to deal with data, which is more standardized, right? When we speak about other types of data, for example, measurement of weight or height, or even clinical abbreviations for different conditions and indicators, we're used to everything being standardized. And here, when we're talking about biological data and this high throughput data that speaks about individual processes within the cell, at a molecular precision, we are going to be facing this additional challenge of a lot of technical noise and a lot of biological variation. So that's what we will try to address in this program. The program is going to talk about data-driven research in this age of high throughput data in life sciences. So we'll look at a lot of these different challenges that we talked about just now. We'll talk about the reference genomes and how additional references and changes to the reference genome and its annotation can help us understand what is really going on. We'll talk about different computer languages, coding languages, and operations that can help us find patterns in data. And we'll learn about effectively launching different functions and different analysis algorithms to extract meaningful information. And we'll also talk about this big challenge of integration of various biological data so that it can be used effectively in research as well as in industry applications like in pharmaceutical industry and biotechnology. So to do that, we'll talk about different aspects of the way the data is generated, the way it could be analyzed from a statistical perspective, and the way we can account for different types of variations in experimental design so that when we are coming to conclusions, we can go back and retrieve all the steps and make sure that the analysis is robust and reproducible. Now, many of these things are going to be applicable universally to any types of omics data. But as you probably will soon discover, handling everything at the same time is going to become overwhelming. So we'll focus on several examples. And instead of speaking about theoretical approaches, we'll try to go deeper into projects and really see how such analysis decisions can impact the results in the context of very specific examples. I'll speak more about the different project examples in just a few minutes. But before that, I want to introduce you to a place where anyone can start learning about bioinformatics. So let's go to a course called Introduction to Bioinformatics. Introduction to Bioinformatics is a course that is available for free on our educational portal. So Bipsa pasted a link in the chat. To access this course, you have to register and log in. For those of you that are part of the program, you should already be logged in. And I suspect that many of you have already looked at this specific course. So once you get to this course, you will see the Introduction to Bioinformatics. Please put in number 1 in the chat box if you have reached this page. This specific course is free, and I will speak about some additional free courses in just a second. And I will also speak about some additional courses that we will leverage in this program, and they are packaged within the program registration price. All right, excellent. So as you reach this page, again, some of you might have already started on this course previously, but I want to briefly show you what these courses offer and how to best utilize them to start with bioinformatics. So this course provides an overview of big data in different applications of bioinformatics. And, okay, so for those of you that are here, please make sure that you are logged in. Once you are logged in, you can log in with your email address, or you can register with your social media. You will be requested to complete your profile, and you will also be able to change your password once you have completed the registration. So once you're here, you click on Take the Course, and that's the way to get started. Now, this is a free course, so anyone can get started with it. And let's just take a look at what this includes. So if you click on the Curriculum tab, you will see that one of the first things is a survey. And so the goal of this educational portal is not only to allow you to learn about bioinformatics and start practicing applying bioinformatics to different data sets, but also for us to learn more about you and connect with you so that we can identify the most efficient way to help you at the place where you are. Now, after you have registered, you will have access to the intro survey, which we ask you to complete so that we can learn more about your location, your level of education, and the organization that you belong to. After you complete that, you will be able to see some examples that show the data-driven discovery paradigm shift, real-life examples in healthcare, and other types of applications of bioinformatics, including environmental science, agriculture, defense, public health, and others. Now, after we go through some of these topics, we will also discuss how to build bioinformatics pipelines, and we'll use the T-BioInfo platform to illustrate and do a demo together so that everyone understands how to get started with bioinformatics and data sets like genomics and transcriptomics. So as I mentioned, this course includes a survey, and in this survey, we're going to be getting this information, reviewing it, and getting back to you. And we will also ask you to evaluate the content. What we've seen is that both students and faculty find this course informative and learn a lot from the variety of types of applications that bioinformatics has and the critical importance for biologists and others engaged in life science research. Now, this course is going to talk about the emergence of big data that changes everything. It will show you some experts that speak about their understanding of bioinformatics and the way that they affect healthcare today. It will also help you learn the terminology and talk about different types of omics data and evaluate your understanding using a quiz. So here's an example of that quiz. Now, in addition to this free course, we also have a number of other free courses on genomics and metagenomics, and all of them will be introduction to. So you will see introduction to genomics, introduction to metagenomics. So anyone that is learning about bioinformatics for the first time or is joining this program for the first time, we encourage you to take some time to explore some of these free resources and start to really understand how you can get started in this journey of bioinformatics. Now, in addition to these free resources, we are going to be leveraging some additional coursework that we have on this portal. And what's important about this portal is that we are going to discuss how the theoretical material that is being discussed could then be converted to a practical application of bioinformatics. So for example, in each one of these courses on transcriptomics, we are going to discuss the process of generating RNA-seq data, analysis of RNA-seq data for processing, where we get a table of when we have a table of expression. Then you will get an introduction to statistical analysis of RNA-seq data, analysis of RNA-seq data using machine learning and data mining, and finally an introduction to single-cell transcriptomics. We will then continue on to explore genomics, starting from introduction to genomics, variant calling in Genomics 1, and downstream analysis of variants in Genomics 2. We'll also explore a variety of courses on epigenetics or epigenomics. Epigenomic data describes different types of DNA methylation patterns, as well as histone modification that can help us understand non-DNA encoded regulation of gene expression. Importantly, we will also touch on metagenomics. When we talk about metagenomics, we can study 1,000 of different microorganisms or millions of microorganisms that live in communities that function and work together to affect every aspect of our life, from immune responses to responses to different treatments, and also the interaction between the microbiome and the different types of pathogens. So we'll talk about all of these different aspects of different types of omics data. Importantly, every course that you complete comes with a certificate of completion. So those of you that are looking to get a certificate, instead of joining the webinar and looking for a certificate, you will be able to complete that course, passing the quizzes and assignments, and then you will be getting the certificate of completion for that particular course. Now, once you get to that end, it's important that the certificate will be generated for your profile. And so you have to make sure that your profile is complete. In addition to some of these resources, we will also talk about the practical application of bioinformatics analysis. So you will see biodata science is a selection of courses that introduce you to important types of analyses using R and Python. And you can also explore an additional portal that we will utilize during this the next few sessions called code.omicslogic.com. So I'll talk about that in a few minutes. But for those of you that are having trouble with the educational portal, I invite you to go to code.omicslogic.com, also a website where you can register for free and start exploring some of the same analyses using coding in R and Python. The way these courses are organized is they provide with a clear outline of what you might expect from joining a course, understanding the terminology, the biological context, the different data formats, and understanding and analysis logic. You will also have topic mapping by engaging activities where you will understand how the different theoretical aspects of bioinformatics are applied directly to the different data sets and in what order and why. Then you will be able to understand how these apply to a very specific data set or project. So you'll be really able to see project examples taken from research publications and known databases that actually show you the specific file formats in the context of a biological question. Finally, you will be able to deploy an analysis pipeline. And the way that the courses are built is, first of all, you learn about them, you practice with a demo, and once you've mastered the practice, you can start applying this to independent projects that you can do on your own. So after you get a certificate of completion of at least several courses that are free or that are included with this program, you will be able to access the practical analysis that is offered on the T-BioInfo platform. But you don't just start with analyzing data without first understanding how to apply it to a very specific project. So that's why we will also talk about a variety of different project examples. Now, in this program, the Introduction to Bioinformatics for Biologists, we will take a few of these examples that are sourced from peer-reviewed journals in high-impact biomedical journals. For example, we will use modeling cancer precision medicine. One of these projects is taken from a publication that was published in Genome Biology called Modeling Precision Treatment of Breast Cancer, and it was published by a company called Genentech, a company that invests heavily in applying bioinformatics and machine learning to large transcriptomic, genomic, epigenomic, and proteomic data sets to understand how different drugs interact and affect different types of tumors in cancer biology. In this particular study, the authors explored the different omics data sets and how they could help predict the effect of different types of therapeutic molecules that range from the standard chemotherapy to more advanced immunological drugs. So to speak a little bit more about how bioinformatics is used in cancer research, I want to invite Dr. Harpreet Kaur. She is going to be participating in this program to help those of you that are interested in learning about bioinformatics, and will share from her experience being an expert in cancer genomics. Harpreet, could you please share a little bit about your background?
Thank you, Ilya. So just let me share my slide.
If you could please speak up.
Let me just share my slides. So hello everyone. My name is Harpreet Kaur. I have a PhD in bioinformatics. My research is on biomarker discovery for diagnosis and prognosis of liver cancer. So what inspired me to study liver cancer is the high mortality rate of this malignancy and the lack of precise biomarker that can help diagnose liver cancer at an early stage. It is sixth major cancer in terms of incidences worldwide and fourth in terms of mortality. Liver cancer is often at times misdiagnosed or detected too late for an effective treatment. Another factor that contributes to its poor outcome for the liver cancer patient is the frequent reoccurrence of this particular tumor. So with these concerns in mind, I framed a number of research objectives with my collaborators to address these issues using bioinformatics. For the first part of this research project, we used large-scale transcriptomics data to identify a diagnostic biomarker panel for hepatocellular carcinoma, which is primary liver malignancy. So in this study, we employed 30 data sets containing nearly 4,000 samples to elucidate transcriptomics biomarker panel for diagnosis and prognosis of liver cancer, implementing multiple statistical and bioinformatics techniques. For the next part of this research project, we use transcriptomics and methylation data to identify biomarker panel for stage classification of hepatocellular carcinoma. Here, we employed TCGA dataset of 350 samples and elucidate 51 features that include 30 RNA transcripts, 21 methylation CpG sites using number of data mining approaches. The naive-Bayes model classified early and late-stage samples of hepatocellular carcinoma with nearly 79% accuracy. For the third part of this research project, we used transcriptomics data to scrutinize diagnostic biomarker panel for cholangiocarcinoma, which is another major liver malignancy. So in this study, we employed 9 data sets containing 350 cholangiocarcinoma, 133 adjacent non-tumor, and 90 hepatocellular carcinoma sample to scrutinize cholangiocarcinoma specific transcriptomic signatures. And here we have employed a number of data mining techniques like extra tree, SVM, random forest, logistic regression, LDA, etc. So in conclusion, we have identified a universal platform independent 3-gene based biomarker that can predict HCC patients with high precision and also possess a significant prognostic potential. Then we identified a 3-gene based cholangiocarcinoma specific transcriptomic signature that are capable to discriminate CCA from SCC and the normal samples. At last, we identified a biomarker panel of 51 features that include 30 RNA transcript, 21 methylation CpG site to predict the hepatocellular carcinoma at an early stage. So I'm looking forward to being your mentor during this introduction to the bioinformatics program and help all of you to learn about data analysis, machine learning, and visualization in application to large data sets used in biomedical research. In this program, I will be speaking about the role of big data that plays in biomedical research that can generate a hypothesis that is then analyzed and used for accuracy improvement and how this process is affecting clinical care. Now, I would like to pass this session back to Ilya. So over to you, Ilya.
Thank you, Harpreet. And let's address some of these questions that are coming up in the chat as we move on. So what's important about what we're doing today is this is just the first session. In this session, we're giving you an overview of what we will cover in greater detail in 6 additional sessions in the Introduction to Bioinformatics program. We will also introduce slowly, step by step, the different types of analyses and the different types of project examples. So we will actually have experts like Dr. Harpreet Kaur and Dr. Mohit Mazumdar, who will be speaking in a moment, help you identify data sets, extract them from publicly available repositories like NCBI, and run analysis that will help you identify biomarkers, just like Harpreet Kaur just explained, so that you can try and do this on your own. Now, in her research, they focused on liver hepatocellular carcinoma, which is a particular type of liver cells that lead to cancer. So we will go through different types of cancers like breast cancer, lung cancer, liver cancer, and you will be able to see how the same types of principles could be used to work with clinical and omics data. This program is just an introduction. And after a few more slides, after we introduce you to the practical tools that we will be utilizing, you will also hear from Dr. Mohit Mazumdar about research projects developed by previous participants of the OmicsLogic training programs that you will be able to follow and do the same as you make use of these different practical resources that we will be describing in a second. Does anyone else have any questions before we move on? Okay, so for those of you that would like to still complete the registration, thank you for posting your email addresses. BIPSA will be emailing everyone details on how to make sure that your registration is complete. And you're welcome to email marketing@pine.bio with any types of questions that you might have about registration or links that are not opening. Okay, so let's continue speaking about some aspects of the program that will help you make more use of bioinformatics and apply clinical thinking to applying bioinformatics to different types of data sets. Okay, and also for those of you that are interested in cancer biology, I think bioinformatics has shown a lot of promise in cancer biology, and we'll talk about a lot of different examples that will help you understand how these methods work in real clinical settings as well. Okay, thank you, Harpreet. All right, so what's important as you join the program is to make sure that you complete your profile. So each one of you gets a personal profile. So for those of you that are interested in doing more with this, the interaction during the session is really to help you understand some basics. But the work starts when you start practicing. So this is not going to be interactive in the sense that 100 people can all talk at the same time. That I don't think is going to be very useful. But what will be useful is when you start applying the methods that you learn to specific data sets, and we start interacting with you around the analysis challenges that you might have to clarify how to apply these tools effectively. So the first step is to really complete your profile. As you work through the different coursework, you will receive assignments, and those assignments include analyzing data, which includes running some bioinformatics pipelines. You will also see that a lot of the courses have quizzes, and those quizzes are tracked so that we can understand where you are failing and try to make sure that we understand and you understand all of the concepts that are being covered. And we also track your progress over time, making sure that you are actively participating and benefiting from the different resources that we have made available. At the end of that process, when everything is finalized and you have gone through the different coursework and started working on an independent project, you will receive a certificate. The certificate could be a certificate of completion or a certificate of excellence. So for those of you that want to distinguish yourselves and really showcase the ability to analyze data, the more you do, the more it will come up in your profile. For example, in this profile, you can see that Sarah Zapico, who is a faculty at University of Florida, has reached over 18,000 points. So anyone who reaches more than 10,000 points will be able to get a certificate of excellence. And also for those of you that complete an independent project will also get a certificate of excellence. Now, let me introduce you briefly to some of the other practical resources that we will leverage during this program. This is another important part of what we will do, actually applying big data analysis to these data sets that we describe about cancer and other types of conditions. So please go with me to server.t-bio.info. Okay, so maybe put in number 1 in the chat as you reach this link, server.t-bio.info. When you join this program, you will get a personal account on this big data analysis platform. For those of you that are joining today as observers, you will be able to use a demo account. So Bipsa, if you could share just the first demo account details with those that are joining us today. Okay, so those of you that already have completed your registration, you should have gotten an email with your personal account. Your personal account contributes points towards your own account progress. And for those of you that do not have an account yet, have not completed your registration, you can use this demo account for now. So again, I see that one person has reached the server. Has anyone else reached this server and used the demo account? Okay, great. So we have a few more. Please let me know. I will give it a pause, just a couple of minutes until everyone reaches. So this is the T-BioInfo platform. It is a big data platform developed by the Tauber Bioinformatics Research Center. This is a research center at the University of Haifa focused on development of novel algorithmic approaches to big data analysis, specifically in application to omics data. You will find that this specific page demonstrates the different areas of analysis, including next-generation sequencing data, mass spectroscopy, structural biology, and tools for data integration and modeling. So what we will do today is look at one example of a bioinformatics pipeline related to transcriptomics. So please open this arrow and go with me to this demo for RNA-seq data. Once you're here, click on Start, and you will see that each button represents an educational resource where you can learn about the typical steps to process or prepare sequencing data for downstream analysis. The first step is to clean up the reads. Next-generation sequencing produces shotgun sequences, which means that they are taken from random places in all of the material that has been shattered, and are sequenced in short reads. These reads could range anywhere between 30 nucleotides and 350 nucleotides in length. They could also be different types of sequencing, including single-end or pair-end. This allows us to increase the accuracy of the sequence that is captured within each read. As you look at the pre-processing steps, you will see that there are a few different options to pre-process this information. After it is processed and it is ready for analysis, we perform mapping. That mapping is taking the short reads and aligning them to the reference genome. After those are aligned, they have to be quantified so that we will be able to analyze the levels of expression for different genes. So let's click on OK, and you will see that some additional steps are becoming available. So for those of you that are following along, now I can connect the Start button with the pre-processing steps. The only available pre-processing step at this point is Trim-O-Matic. Trim-O-Matic is a mapping, is a tool for mapping the reads to a set of references that include technical amplification and library preparation sequences that will clean up and reveal only the accurate read information that will be used for downstream alignment and quantification. After you click on OK, you can now click on PCR Clean. PCR Clean addresses the issue of bridge PCR amplification, which can be biased and can affect highly expressed genes. After I'm done with the pre-processing steps, I have a couple more options. One option is to use mapping on genome to identify different types of alternative splicing events. Or if I'm not interested in alternative splicing and I'm interested in gene expression, I can click on bow tie to T, which will map those reads on the transcriptome. So only the expressed genes that are recorded in the GTF file. Again, we will review all of these steps in greater detail in our next session, and they are also covered in Transcriptomics 1. So once I've connected the pre-processing steps with mapping on transcripts, I can now quantify the gene expression by producing a table of expression in read count, FPKM, which is fragment per kilobase per million, or TPM, transcript per million. Now the pipeline is complete. I can click on End and look at the output. So here's the output. These are what's called table of expression. And table of expression could be for genes, isoforms, or exons. And you can also see an example of a cell line expression data normalized and annotated, as well as some of the clinical subtypes included in this particular data set. So this is a quick example of what's called building a bioinformatics pipeline. So typically, if you are a bioinformatician, you would have to launch different scripts like PCR clean or Trim-O-Matic, connect to different databases. For example, Trim-O-Matic uses a database of technical sequences, or load in your reference genome and the annotation file called the GTF file to eventually make sure that the file structure goes from one step to the next and produces the final output, as well as many intermediate steps. You will discover very quickly that this is a very intensive process that requires high CPU power and a lot of RAM and storage. So for those of you that are going to be joining the program, we will review in detail how to build such pipelines and also look behind the scenes on how to apply different types of R packages and Python packages to perform what's called downstream analysis. So on the T-BioInfo platform, it is built for research tasks that require big data processing and high performance computing. After the data is processed, after you have received the table and you can start analyzing the data, you can turn to statistical analysis, visualization, and machine learning. So what we will do is go through different types of pipelines for different types of omics data, and then extract the data that the pipeline has produced to start applying these packages using coding. What's important here is that biologists and clinicians are probably not as interested in developing proficiency in coding in R and Python. But it is important for all of you to understand what goes into coding and how coding packages and libraries are typically used to analyze, visualize, and share the results between participants of the wet lab and the dry lab. So we have included a number of courses that you will be able to go through, including analysis of sequencing data, integration of different types of data using statistical analysis, machine learning, different types of data mining or unsupervised machine learning and supervised machine learning, and also annotation. So before we move on, I want to ask everyone if you have any questions about what we covered, and I'll look through the chat to make sure that I haven't missed anything. Okay, again, I see that there are some questions about registration, pricing, how to register, how to access different resources. I think the best way to do that is to register for the full program, and we will work with you individually to make sure that you understand how to access and also how to leverage some of these resources in a way that you learn about bioinformatics and learn to apply them to your own project. And we will definitely be emailing you the links. Requirements for your own computer: you don't actually have to have anything more advanced than a Chromebook. If you can download and upload files, that's enough. So you can actually process the data sets on the cloud. So those are going to be the big data sets that you process. And then you can use all of the different types of analysis that we're discussing here on the cloud. So you don't actually have to do any of it on your computer. You will benefit from doing some of it on your computer. So it's beneficial to have Excel or Google Sheets, and also to be able to upload and download files. So I don't think that an Android phone is going to be the best option. It's much better to have a laptop, at least a Chromebook. Okay, and we've captured all of your email addresses. So those that have shown an interest, you could actually, you will receive an email from marketing@PineBio. Cloud processing, does it need good internet speed? Yes, good internet speed and reliable internet speed is going to be beneficial. If you can join this session and see everything that we are doing in this session, that means that you have good internet speed enough for what we are doing. Okay, so before we continue, we are going to give you an assignment. The assignment is, first of all, complete your profile on edu.T-BioInfo. So make sure that not only you register, but you put a profile image, you write a little bit about yourself, maybe add some social media links so that people can understand who you are. Join our social media groups, which we'll talk about in a second, and make sure that your profile is complete. Then you can also explore using this demo account, or if you already received your full account, you can explore the server.T-BioInfo and code.OmicsLogic.com, some effective practical tutorials that we will be doing throughout this program. And please complete the Introduction to Bioinformatics course, fill out the survey, and try passing the quiz. So that will give us a good sense of who you are and how you are able to apply the understanding of bioinformatics to different types of projects. Can we run the R scripts in RStudio? Yes. All of the scripts in the OmicsLogic coding platform are downloadable. You can just copy them and open them in RStudio, and we will actually be sharing the R scripts with you also from the pipeline itself. So now to continue speaking about projects, I see that many of you are interested in cancer. Some of you potentially might be interested in other types of projects, infectious diseases, and others. I want to introduce you to Dr. Mohit Mazumdar. He is responsible for partnerships and is a project mentor in our OmicsLogic programs. Mohit, please take it over. One second, Mohit needs to be, okay, you are co-host.
Ilya, co-host is me, so you have to make Mohit co-host.
I did. Oh, that's you. One sec. Okay.
Okay, thank you, Ilya. And hello, everyone. So let me just share. So my name is Mohit Mazumdar, and I am one of the global representatives of Pine Biotech. My PhD was focused on developing a novel method for sequence analysis using machine learning algorithms. After working on several bioinformatics research projects, I joined bioinformatics industry, where I'm collaborating and working with the academia and healthcare industry and the pharma industry to provide support for bioinformatics research and training. But it's really exciting to be a part of this OmicsLogic experience as a project mentor, and to be able to work on translational research with a diverse group of students and with diverse expertise. More often than not, we hear about the biomedical data being used by pharma industry and publications from top research labs. So as Ilya mentioned, there has been a remarkable shift over the last decade in the cost of generating molecular measurements. So most impacted by this change is in the area of genomic medicine. We heard about different type of cancers. where it is now possible to move from generating, reference or population level data to a, individual level data. So now we are collecting that sort of information, and concurrently there has been advances in development of new algorithms and tools to integrate and interpret this data, which is what you saw the T-BioInfo platform as an example as well. So if you see this survey, this latest survey, you will find out that AI and big data have the biggest impact on the pharmaceutical industry. However, to understand this application and to be able to find information from this big data, it's important to learn about the analysis strategy and get experience working on real data sets. So together with our collaborators from top universities, we have prepared tutorials for you that takes you through a project-based approach, where we take data sets from high impact publications and use them for practical exercises. Recently, we finished a few projects collaborating with students, researchers, and so recently, as I was telling you, that we finished a few projects collaborating with students. So all of these projects and applications were proposed by the participants. So here is a picture of, if I don't know if my animation is seen or not. So there are few participants, for example, Anurag Kines, who collaborated with Avra Bohra, and they worked on the infectious disease project, and people published on different platforms like LinkedIn, and they got the recognition of that project being finished and contribution in that research area. So for example, here Urja Parikh recently worked on her research fellowship and her project on glioblastoma stem cells, and now she's continuing her research. So this is a link. Let me share this, and maybe let's go to this link. So let me again share the link. Okay, so this is the page when you get to the project examples, which I've just, which is being just shared, I guess.
Dr. Mohit, it's shared in the
Yes.
chat.
Thanks, Viksha. So this is where you will find more relevant examples. So here we talked about the guidelines. How do we go about a big data bioinformatics project after you get the learning? And we talk about poster publication or going towards a full-fledged publication. How do we reach a place where we have a clear methodology defined in terms of abstract, methods, results, and conclusion? There are some examples that we did recently in collaboration with LBRN. So you will find those as well, and you will find the abstract and learn from examples. So some of the participants from the infectious disease program and research fellowship program collaborated in a group. So we want people and this research groups to collaborate so that they can speed up this process of working on research, and different people have different expertise. So that helps in different aspects of the project. So this is such an example. So after the project is finished, or it reaches a space where there is more conclusive information, the project is being recorded as a video. So you'll find videos also where the participants are describing the entire project and what is the outcome of this project. So here you will see research on SARS-CoV-2 as a part of infectious disease research project. And then you'll find the research project that I just talked about glioblastoma stem cells, and the data available. This was all done through the publicly available data sets. So you will find them in different repositories that we discussed today. So there was an interesting, research proposals that we were evaluated and worked on during this entire summer and fall, where people, our participants and researchers and faculty and research labs. So we collaborated with various research labs and faculties that came up with their own ideas find another example of from Sarah. So Sarah Zapico. So you saw that the certification as well. So this is the translational aspect of your learning. So please look at the link. And here, I mean, interestingly, the project-based approach or the OmicsLogic approach has been taken really well by undergraduates and high school students. So we have Hannah Davis here. She is a high school student and finished her bioinformatics project working on our platforms. You'll find more examples from high school students that those recently joined our genomics in the virtual lab program were able to work on their own research project on cystic fibrosis, and another example from that program. So you will find the translational aspect and what is the outcome, one of the outcomes of these research projects by looking at some of these examples. So with this, I think I can pass it on to Ms. Deeksha. She'll be able to guide you with other aspects like program registration and access to various resources that we discussed today. Thank you, everyone.
Thank you, Dr. Mohit. Pallav, if you have any question, please put them down in the chat box so that we can look at it and get back to you. So this is the program registration page. I have already shared the link in the chats, and I will be posting the link again for everyone. It's called bioinformatics.omicslogic.com. So if you scroll down to the site, you will notice that all the different details of the session are added here. And you'll be able to read some reviews. And I'm doing this one more time so that all those participants who have joined us for the first time today are able to complete the registration. And to complete your registration, the process is very simple. You either click on Get Started or on Pricing. Either way, this is the thing that pops up. And you can choose between 3 options here. So the beginner option will give you access to all of the resources that were discussed in this session. The intermediate membership gives you access to all the resources plus a one-on-one mentor support throughout the length of this 1-month program, while the advanced membership gives you access to the beginner courses, projects, the OmicsLogic platform and the T-BioInfo platform, along with mentor support, as well as extra additional 30 more days to work on your project. So that becomes like a 2-month program for you. So you get not only just 1 month, another additional month to work on your project. So this you can choose according to your own preferences and choices. And for those who are mentioning that they would want to do projects, please leave any kind of requests that you have regarding projects to marketing@pine.bio, and we will be reaching out to you with your requests. And now moving on. So once you are registered in this platform, so if you scroll down on this pop-up, you'll see that there is payment details. The advanced courses would require you to know the basics of biology as well. So I will be talking about some of the upcoming advanced courses very soon. So just to talk about the Introduction to Bioinformatics program, which we are starting today. The next thing, once you complete your registration, what you receive is your own profile. So here I have Dr. Prashant Mishra's profile. He is part of this program. So you can see that he has a badge which determines that he's part of this membership. He has been doing some of these courses and taking part in the coding platform, which is a code.omicslogic, and he finished some of the courses here. So you see the points that he has gained. So this is how a completed profile looks like. It has a profile image and a profile name. Again, you can contact us marketing@pine.bio for any kind of queries related to any personalized projects. And once you finish your profile, the next thing that you have access to is the organization page. This is the organization page exclusively available only to mentors. And You will be able to see details about the program, see the different syllabuses. So today we are on this first session on 13th October. Our next session will be on 15th October. And also if you scroll down below, you will be able to see all the different participants who are joining together with you and their performance. So we can track everyone's activity. And not only this organization page will give you access to courses that are part of this program. So as Ilya was mentioning, you should be completing, this is for everybody, even if you're not part of this program, you can complete Introduction to Bioinformatics, but you also get access to other premium resources like Transcriptomics 1, 2, then you have Introduction to Epigenetics, and more are added as we go along. We also offer projects as part of this training. So these projects are available free to our participants who are part of this training program again. And these are asynchronous projects which help you in your own learning about how to get started with a bioinformatics project. So these are the other resources that are available to you. And we have a progress tab to track everyone's activity. So as we are starting today, we see that the activity is just starting out. So as we move on, we will have more graphs going around. So in a way, we are able to track anybody's progress and help you go forward. So even though it will be a group program where a lot of people are joining from different fields of life sciences, we are able to help each other and guide everyone throughout this program using this organization page and our forum. Yes, the free courses, Siddesh, also come with individual certificates. So once you complete a free course, it will be automatically generated. Not only just the free courses, also all of these courses will each carry their own certificate. So you not only earn a complete program certificate, but you also earn one certificate for doing each of these courses. Okay, so that is there. And you can also see the syllabus on this tab. Okay, Sheetal, I will be emailing everyone the links to the free courses after this session so that you can look at them again. But again, if you are on the Edu platform, you can just go to Introduction to Bioinformatics and start taking the free course from here. This is the first free course, as Ilya has been mentioning, which is available for you and open to everyone. So you can take this Introduction to Bioinformatics course. The third thing that you have access to is the T-BioInfo platform. So this we already had a demo section about, and every participant will get access to the educational access to this platform. This platform is also available as a research tool that can be used by universities, research labs as well, and individual researchers. But for program participants, they will have access to 5 terabytes of data in the space in the cloud. So, and they will be able to not only work demo pipelines, but also some pipelines which are shared with you. And you will also have access to the code OmicsLogic platform. Now, for all of you who may be interested in advanced courses, we have several of them starting in the coming months. So somebody was interested in breast cancer and transcriptomics. So we have this transcriptomics course starting in December. We also have a program on infectious diseases starting on 9th November. And data science is already ongoing, but it's still open for registration through this week. It has started yesterday on 12th October. And we also have another program coming up on metagenomics. So this is the entire list of the programs that are available for this year. For those of you who are interested in specific areas which are not covered under the program,
yeah, Lisa, I would like to address some of the questions that have been coming up.
Yes,
in the
sure,
chat.
Dr. Mohit. I just wanted to show the research fellowship page. Just one moment. You can actually start talking about this as well.
So start with Dr. Sheikh Sheikh Bharat from Chennai. So my area of interest is prenatal and cancer genetics. I want to I want to I want the details of NGS data analysis and bioinformatics application course. So Dr. Sheikh, we would start this introduction to bioinformatics course, which will cover all of these aspects that you have talked about. But talking about your area of interest is more towards a research project. I see there are other questions similarly on different research areas, such as forensic sciences and nutrigenomics. So once you learn about the application of the technology, you can use it for all these research areas, finding data sets relevant to these research areas. So the important thing is to understand the application, like transcriptomics, how to analyze transcriptomics data, genomics, how to analyze genomics data, and what you can find out of that genomic data. So if you already have a research question, so that actually helps. So you can look for data sets that are, kind of helps in finding those answers. So that is the kind of data-driven research we are going to do in many of these programs. And that's what our focus is, to simplify bioinformatics and big data research, because doing this without the infrastructure is really a challenge that we discussed and we know about. So this platform and entire learning experience gives you that opportunity to be able to work on some of those aspects that you really want to using the big data. So there is no certification for this individual session, but you will get certificates once you finish the asynchronous course that we shared. So the asynchronous course are Introduction to Bioinformatics, Introduction to Genomics, and Introduction to Metagenomics. So these all courses are going to give you an overview, like the question to the application of bioinformatics and forensic sciences. So Introduction to Bioinformatics, please look at that course and try to finish that course. It's a very simple course. And Introduction to Genomics gives you a direction that what is all going on in genomics, what we learn in genomics. And then there is metagenomics. So these are free courses that are available and that you can gain certificate from after you finish those courses. So the question to answer Shah. So Shah, please email me. Email Beecha the research area and the research that you are interested in. And bioinformatics for neurodegenerative diseases. So we have Julia Panoff. She's a neuroscientist and bioinformatician working on neuroscience. So one of our interns and few of our interns were working on neuroscience and data sets associated with that, and some of them worked on Alzheimer's diseases. So we also have a project on Alzheimer's diseases. So you can work in neurodegenerative diseases if you register for research fellowship. So that is another program that is available for researchers who are pretty much aware of this NGS technology. Okay, Santya, we will send you the details of the research. This is the research fellowship program. Yes, thank you, Biksha. So bioinformatics, machine learning, prerequisite knowledge. No, this is where you can learn, Joshua, bioinformatics and machine learning and application of machine learning in big data bioinformatics, especially because bioinformatics is a really big field because of its application, from structural data to genomic data to different kind of data sets. So this is about NGS data, and NGS data you can learn to apply machine learning with this course, introductory course also, and the associated material. So there's a lot of asynchronous material that guides you through how do you do NGS, how do you apply gene expression, machine learning on gene expression data. So all those aspects are very simplified and easy to learn through this platform. That's what I have experienced. So yeah. So internships are provided as a research fellowship program right now, and internships are and an advanced internship is only provided for those who are expert in our platform. So you have to go through an OmicsLogic platform, and you have to show that progress in your research, in your learning, to be able to get any kind of internship, long-term internship or a short-term internship. But the research fellowship program is a program where you can work on your project, and you have to only take the license for the T-BioInfo platform. Currently, we are supporting the research fellowship program ourselves. So just you have to get the license, and the sessions and OmicsLogic training and everything is included in that.
Thank you, Dr. Mohit, for answering the questions in the chats. Yes, so everybody who has left us an individual comment on the different areas of analysis and the different research areas that you're interested in, or the different programs, please wait to hear from us, because we will be individually looking into these requests. And even to Shah, if you had sent us your email ID or sent us an email to us or left your email ID in the chat, please wait to hear from us. We will be reaching out to everyone so that you can do something or take up any of these courses or do a research project with us which you are interested in. So thank you again for joining today's session. Saying that, if you are joining this program, and the next date for the second session of this program is October 15th, which is going to be exclusively available only to our participants. And you can join via this link. Okay, Deepak, we will be sharing the fellowship link again personally to everybody via email. So thank you again for joining today. So if there are no more questions, I would like to end today's session. Thank you, everyone.
Thank you, everyone.
Thank you, everyone.
Okay, if you're still leaving your emails in the chat, and if you have left them before, we have a note of all of your email IDs and your personal requests. So we will be reaching out to you. And you can also request or send us any information or an email to marketing@pine.bio for any other queries you may have, especially if you have a research-based query or a program query and you would like more clarification on it. So thank you so much to our speakers today and to our participants. We'll see you again soon.